Okay, so I'm going to talk about blood typing today, but first I want to talk about blood donation and blood use. So as you can imagine, blood is such a valuable medical commodity that most oftentimes its components parts are separated. Well, first of all, I should say they're either donated individually or they're separated after collection. So we separate it into the plasma, the white blood cell platelet layer, and the red blood cell layer down below that. And then we can use these different components individually. So plasma. Here's a bag of plasma there. What do we use plasma for? Well, plasma contains clotting factors. And as you guys know, clotting factors are necessary for the cessation of bleeding for hemostasis. So, for instance, in hemophilia is a medical condition where you're missing one of the clotting factors, and therefore you have an inability to form clots. And so plasma, which again contains these clotting factors in it, are very valuable to hemophilia patients. What about the platelets? As you guys know, platelets are also involved in hemostasis. They form a platelet plug. And you can derive platelets two ways. One, you can obtain it from the separated blood that we talked about, but there's also something called platelet pheresis, and this is where you can actually they take blood, they separate the platelets, and then they return the red blood cells in the plasma to you. So if you're just taking platelets, you know it's a good way to do it because you don't lose those other formed elements as well as the plasma. So why are platelets important? Again, they're involved in forming a platelet plug, and let's see. So people who have undergone a bone marrow transplant, cancer patients, leukemia patients, all benefit significantly from platelets. For example, if a cancer patient is undergoing chemotherapy, one of the side effects is a low platelet count. And so again, they have these issues with potentially having life-threatening bleeding situations. And so platelets are very valuable to these individuals here to help stop bleeding. And then we get to the packed red blood cells, and they're called the packed red blood cells because they're all packed down there. And that's all we have. This is only packed red blood cells in there. There's not plasma, there's not white blood cells, anything like that. So to collect red blood cells, if you give a whole blood donation, then they can separate the red blood cells like so. There's also a method called power red that collects red blood cells while returning most of the other components to the blood. So what do we do with packed red blood cells? Well, they're used to treat hemorrhage, anemia, and basically improve oxygen delivery to the tissues. We talked about anemia yesterday, a low red blood cell count or a low hemoglobin count, and the consequence of that is you have low oxygen carrying capacity. So packed red blood cells are certainly used to treat anemia. All right, so blood transfusion. What is a blood transfusion? A blood transfusion is the process of receiving blood products, this is the recipient here, into one's circulation intravenously. And you can see this individual is getting this blood from the arm, into a vein on the arm. So here we have our blood bag. This is the donor, and then you can see, it comes down here, and this is the recipient. So safe blood transfusions depend on careful blood typing of the donor. So this is the donor right here in the blood bag, and the recipient. And the reason this is important is because we want to prevent an immune response in the recipient. We want to protect the recipient. And so basically what it's used to, blood typing is used for, is to check transfusion compatibility, the compatibility of the blood from the donor and the blood in the recipient. The blood group that we're going to deal with here is the ABO blood group. And there's like 35 total blood group systems around the world. The ABO blood group is the most common human blood group system. And within this blood group system, we have blood types A, B, AB, and O. Now, one's ABO blood type is determined by the hereditary presence or absence of specific antigens on the red blood cells. And in particular, what antigens we're looking for antigens A, and we're looking for antigens B. So here's a red blood, let me get a different color. Here's a red blood cell that I'm coloring blue. And attached to this red blood cell, let's go greenish, is an antigen. That's an antigen right there. And what antigens are molecules on the surface of all cells. Okay, all cells within our body, on bacterial cells, all that kind of stuff. And what they do is they enable the body to distinguish its own cells from foreign. So we use these antigens, the immune system uses these antigens to make sure that the cells in the body are cells of our own, from our own self, and they're not foreign. So in that same regard, these antigens are molecules that are capable of inducing an immune response. If you have an antigen in your body that's not your own antigen, then the immune system is going to recognize that, and it's going to cause an immune response. So these antigens here, they're part sugar, part amino acid, and this particular one here is this particular structure is called an A antigen. And the presence of an antigen names the blood type. So if you have an A antigen on your red blood cell, then you're going to be type A, or at least type A. Here we have a B antigen on the red blood cell, so that makes this individual type B blood. If you have both an A antigen and a B antigen on the red blood cell, then you're type AB. And as you can imagine, if you don't have any of these, if you don't have colors back here, if you don't have an A antigen or you don't have a B antigen on your red blood cell, then you're type O. So the presence of the antigen names the blood type. All right, so let's look at this table here. These are the different ABO blood groups. These are the blood groups right here. So we have A, B, A, and O. These are the antigens that we find on each of those red blood cells. So not surprising that on blood groups AB, we find antigens AB. On blood group B, we find B antigens. Okay, so all this should make sense now. The next thing that we need to look at are antibodies. Antibodies that we find in the blood of these particular individuals. And what an antibody is, an antibody is a large Y-shaped protein produced by the immune cells that are used to neutralize pathogens such as bacteria, virus, and other foreign material, including foreign red blood cells. So the reason we're going to look at this, the antibodies that are present in each of these individuals, is because blood transfusion compatibility is a matter of interaction between the antigens on the donor red blood cells, so these antigens on the donor red blood cells, and the antibodies in the recipient's plasma. So antigens on the donor's red blood cell and antibodies in the recipient's plasma. And the relevant antibodies that we need to consider, there's two of them. One is called anti-A antibody. And guess what anti-A antibody does? It recognizes A antigens. A antigens are foreign to them and it recognizes it. We also have anti-B antibodies. And anti-B antibodies recognize B antigens. Okay, so the next thing I want to do here, change my colors. There we go. The next thing I want to do here is go through each of these different blood types here and look at the different antibodies they have and ask ourselves, why does each blood type have the antibodies that they do? Let's see. Let's start off with the AB blood group. Why does the AB blood group? They don't have any of these relevant antibodies. They do not have anti-A, or they do not have anti-B. Why is it the case that a AB blood type individual does not have any of these antibodies? The answer to that is, if they did have these antibodies, if they had anti-A present, then it would recognize this A antigen and cause an immune response within the body, and we don't want that. Same thing with anti-B. If this individual had anti-B, then it would recognize these B antigens, cause an immune response, and we don't want that. Let's look at a type B individual. Type B individuals have anti-A antibodies. First of all, why do they not have anti-B antibodies? Well, we just answered that question, because if they had anti-B, it would recognize these B antigens there, cause an immune response, and we don't want that. So we know they don't have anti-B. Why do they have anti-A? Here you can see this individual has anti-A antibodies in there. And the reason that's the case is because the red blood cells do not have A antigens on it, which means that A antigens are foreign. So these are looking for A antigens on red blood cells, because the A antigens are foreign to this individual, because this is a type B individual. Same principle applies to type A. Type A does not have anti-A antibodies, because it would recognize its own antigens. What it does have is anti-B. So we have these anti-B antibodies that are floating around in the plasma, and what they're looking for are B antigens, because B antigens are foreign to them. Lastly, we have type O. And type O has both anti-A, anti-B. And why is that the case? Well, that's the case because their red blood cells don't have A or B. So if you have a red blood cell that comes into the picture and it has an A antigen on it, or it has a B antigen on it, then that's considered foreign. So they have both of these antibodies to recognize both of these foreign antigens. Okay, so let's get into some of the blood transfusion stuff. My question to you guys is, can type B blood be given to a type A individual? Type B blood given to a type A individual. And I want to point something out. If this is the donor here, so this is type B blood is the donor. When you give this type B blood, you're only giving packed red blood cells. You're not giving any of the antibodies that are present in this type B individual. Remember, you're taking the red blood cells from an individual, and this is all you're taking. When you spin down blood, antibodies get caught up in the plasma in there, and we're not donating that. We're only donating the red blood cells. So we're not donating these antibodies along with the red blood cells. So let's get back to the question. Can these type B blood cells be given to a type A individual? And the answer to that is no. That would be a mismatched transfusion, because the type A individual has these anti-B antibodies that would recognize the antigen and cause an immune response, and we don't want that. And that's what this is showing right here. If we have a type B donor, you give that blood to a type A recipient, then these anti-B antibodies are going to recognize these antigens. And what that does is it causes something called an agglutination reaction. Basically what's happening is these antibodies are taking these red blood cells and they're clumping them up together. They're immobilizing them. So these are the B, we can call these the B antigens here. And what happens is we have these anti-B antibodies that bind to them, and it basically clumps these red blood cells together. It agglutinates them, okay, and it immobilizes them. And then what's going to happen after they're immobilized is the immune system's going to come along and it's just going to destroy all of this. So if you have a mismatched transfusion and you have the immune system agglutinating and breaking down these red blood cells, it can be a real problem. So here we have a mismatched transfusion. In this case, we have a type A donor giving to a type B recipient, and that's a mismatched transfusion. You get an agglutination reaction, and this is the agglutination reaction right there where all these red blood cells are clumping together. There's another one right there. And the problem with that is that these red blood cells can get lodged in these small blood vessels. And what does that do to blood flow downstream of the blood vessel? It cuts it off, so it's no longer getting oxygen. And that's a problem, as you can imagine. Okay, so this is something that you guys can look at and work on, and we'll go over it in class before we do the blood typing lab. But here we have our blood groups. These are the antigens on there. There's the antibodies, so you guys understand all of that. And what I want you guys to do is for each individual, I want you guys to go down and tell me what blood types each individual can receive blood from. Here's a type AB individual. What blood types can an AB individual receive blood from? Can they receive blood from an A individual, B, O, AB? Those are the questions that I want you guys to ask. And again, it's a matter of interaction between the antigens on the donor red blood cells and the antibodies that are present within the recipient. So go ahead and do that for all of them. And after you're done with that, you can tell me which blood type is the universal recipient and also which blood type is the universal donor. These are the answers here that we will reveal. This is some interesting frequency of the different blood types in the U.S. population. Whether you're white, black, Asian, Hispanic, or Native American, the most common blood type in each of these cases is type O blood. But there certainly are some differences between the frequency of these blood types within these different populations, or within these different races, I should say. Now something's missing here. Something's missing from our blood type. If you've ever gotten your blood type assessed, then you know that we're missing something. And what we're missing is either this little plus or the minus on the end of our blood types. And whether you have a plus or minus is based on another blood group. It's called the RH blood group. And the principal antigens in this group are C, D, and E, but by far the most active one is D. So if you have a D antigen on your red blood cells, and that's what this crazy little antigen there is representing, then you're considered RH positive. If you do not have the D antigen, then you're considered RH negative. And what you do is you combine the RH blood type, the positive or the negative, with the ABO group into a single expression. So type O positive. What does the positive mean? The positive means that we have a D antigen. AB negative. What does the negative mean? The negative means that we do not have the D antigen on there. 85% of the U.S. population is positive. Okay, so unlike antibodies of the ABO group, the anti-A and anti-B, the RH antibodies which is anti-D, are not normally present in the blood. So I'll take a step back there. The antibodies associated with the ABO group, anti-A and anti-B, those are present in the blood initially. Okay, those are always circulating around in the blood, looking for improper blood cells in the blood. That's not the case, I should say, with what I imagine is most antibodies, and also with this anti-D antibody. These are not normally present in the blood, and instead it takes exposure to the antigen before the body produces them. So let's look at a scenario here. Here's an Rh negative individual. If an Rh negative individual receives an Rh positive transfusion, so they receive red blood cells that have this D antigen on it, the producin, this individual right here, is going to begin to produce anti-D antibodies to attack these D antigens there. But the first time you're exposed to an antigen and you produce antibodies for it, you don't produce that much, okay? And that's what this is looking at here, and we'll come back to this with the immune system. But let's see. Yeah, so here, this is the first exposure, and we'll just use the scenario we're dealing with. So this would be your exposure to a D antigen. And if you're exposed to a D antigen, then it takes a little while to produce those anti-D antibodies. And even when you do, you don't produce, a huge amount of them. So if you have if you have a blood transfusion and you're Rh negative and you have a blood transfusion with a positive individual, you will get a response, but it's not a very dangerous response. Okay, so there's little danger if you get a mismatched transfusion the first time from an Rh positive individual. The danger, however, is with the second transfusion, because the body has something called immunological memory. So this is the first exposure, and this would be the second exposure to that same antigen. And I should say what we're looking at here, this is antibody concentration. The second time you're exposed to something, because of that immunological memory, the response is much more rapid and it's much greater. You're producing a much higher level of antibodies. And so that's when you get that serious agglutination, and you can have health problems because of it. So this same principle is related to something called hemolytic disease of the newborn. And if I'm sure there's a few mothers out there, then you're probably familiar with this scenario here. So in this scenario, we have an Rh negative mother, and this is the first pregnancy. And so here we are here. So if an Rh negative woman is present with an Rh positive fetus, so here we have an Rh positive fetus, all of these red blood cells have these D antigens on them. So these are the D antigens. And these are in the fetus. So if an Rh negative mother is pregnant with an Rh positive fetus, then she may become exposed to these D antigens here during the bleeding that occurs when the placenta detaches from the uterus at birth. So at birth, when the placenta detaches from the uterus, some of these D antigens can cross over into the mother's bloodstream. If that's the case, then she's going to start producing anti-D antibodies as a response. Okay? But it takes a while to do that. And so this baby, this fetus here, is safe. The problem is if she becomes pregnant a second time with an Rh positive fetus. And the problem with that is now she has these anti-D antibodies that can cross over the barrier into the fetus's blood supply, and it can agglutinate the blood in the fetus. And as you imagine, that can be a serious thing, and that's called hemolytic disease of the newborn. Now they do have ways to prevent it, and the name of the drug that they give is Rogam, and I think they just give this during some phase during pregnancy just to prevent this whole thing from happening. But basically, this is what happens. You can read. Let me see. Yeah, so what this is these are a bunch of anti-D antibodies. And what they do, they must be inactive anti-D antibodies. What they do is they recognize D antigens on the baby's red blood cell, which prevents the mother's own D antigens, active D antigens, from binding to it and agglutinating it, I believe is what happens. Okay, and this blood typing material, we're gonna do a lab about it. There's going to be a little on the lab exam, but this is not going to be on the class exam. Instead, there's going to be a little quiz that I'm going to post right now, 5-point quiz, and that's how you guys are going to be responsible for this material.